Our next topic is the topic of proteins. And proteins are an amazing molecule in that they essentially carry out all of the functions and do all the reactions of living things. And without proteins, we would not be able to survive. Proteins include a number of different types of molecules, maybe most importantly, enzymes. And we'll talk a little about enzymes a little bit later on in the course. But for this video, we'll talk about proteins and what are they, how are they formed, and what are some of their functions, their jobs. And this is for section B1.2. Proteins are constructed from simple building blocks, monomers called amino acids. And all amino acids have the same basic backbone or structure. That's a nitrogen with two hydrogens, an amine group with a carbon in the middle that has a hydrogen and an R group, and then lastly a carboxylic group, which is a carbon, has a double bonded oxygen and a hydroxide, an OH attached to it. What makes each amino acid different is what's attached at the R group. And it could be as simple as a hydrogen, it could be a carbon with three hydrogens attached to it, a number of different possibilities, but that's what makes each amino acid different. And there's approximately about 500 known amino acids. 20 of those are used to make proteins, are essential for us, for living things. About 20 amino acids are used in building proteins, and this is the overall general structure of amino acids. And so, as we'll see, these get put together in order to be able to make a protein. So to form a protein, amino acids get put together, and this forms a dipeptide. When two of these are put together, it forms a dipeptide. This is the linking of two amino acids by a condensation reaction. So just like we saw with carbohydrates and with lipids, condensation reaction takes place in which the OH from the carboxylic group and the hydrogen from the amine group are removed. Because water is being removed, dehydration synthesis, that gets removed and a bond forms between the nitrogen and the carbon, and that bond is called a peptide bond. When there's two amino acids that are linked together, this is called a dipeptide. If it's 20 amino acids, it's called an oligonucleotide, and if it's more than 20, it becomes a polypeptide. And this is a polypeptide is the main component of what makes up proteins. Where proteins are made are at the organelle called a ribosome. And ribosomes can be found in two different locations within the cell, either free-floating ribosomes within the cytoplasm, and typically the proteins that are made at these free-floating ribosomes are going to be used within the cell, so those proteins are used inside of the cell. The other location that ribosomes can be found, as we've discussed, are attached to the endoplasmic reticulum. It makes it rough endoplasmic reticulum, and oftentimes, not always, but oftentimes these proteins that are produced at the endoplasmic reticulum ribosomes are exported outside of the cell and used outside of the cell. Amino acids linked by peptide reactions, this takes place through condensation reaction as we've discussed, and so this is how those amino acids would get connected with one another. Life shares 20 amino acids that are ideal for making a wide range of proteins, and these are the amino acids that are needed by living things. Essential amino acids are ones that cannot be synthesized in sufficient quantities by the organism. So it's essential that they are acquired from somewhere else. Non-essential amino acids can be synthesized through metabolic reactions and pathways, and we'll look at some examples of metabolic pathways in the future. 9 of the 20 are essential to humans, meaning that we have to acquire them by eating something else. And humans and other animals obtain these essential amino acids by eating food. Plants obviously are not consuming food, but they do uptake inorganic nitrogen. It's taken up by the root system, and it's incorporated into glutamine and glutamates through primary nitrogen assimilation, which essentially means that they're taking in the nitrogen and changing the form of it, and then that can be used to synthesize other amino acids and nitrogen compounds. And so this is how plants then can produce those different amino acids. Animal-based foods, fish, meats, milk, eggs, dairy products, for example, have a balance of amino acids that are needed by humans. So if you consume those animal products, then you're acquiring all of the essential amino acids that are needed. Plant-only diets, specifically vegan, it's a little bit more difficult because not all plants contain the necessary balance of those essential amino acids. And so, for example, cereals like wheat, oats, they have a low lysine, the amino acid lysine. There's a low amount of lysine content within those. Peas and beans are low in methionine, and so it's important to have a balance between which plants and vegetables you're consuming in order to make sure that all essential amino acids are being consumed. Modern plant-based diets do provide a balance. It's totally fine to be vegetarian or vegan and still acquire all of the amino acids that are necessary. It's just important to be mindful because they're not widely distributed in all plant products even. Because life shares these 20 amino acids, they're really ideal for making a wide range of proteins. As we discussed, ribosomes connect those amino acids to form proteins, and the sequence of those amino acids, so which amino acids get put together in which order, those instructions come from the genetic code. We briefly talked about that with our DNA very beginning of the course, and we'll discuss how that actually takes place a little bit later on in the course. This provides for a really totally endless possibility of how those amino acids get put together. And so for a dipeptide, if we link 2 amino acids, there's 20 times 20 possibilities because there's 20 amino acids. And so if you take one and connect it with another one, there's 20 times 20 different possibilities. For a tripeptide, so 3 amino acids, it's 20 times 20 times 20. And this goes on for 20 to the nth possibility. Most proteins have thousands of amino acids put together to make up that protein. And so if you think about how many different possibilities there can be, it's essentially endless. There's an infinite number of possibilities given that a polypeptide can be tens of thousands of amino acids. So there's a wide diversity of proteins that can be produced from this diversity of amino acids. The structure of a protein is stabilized by bonds and interactions between the R groups of amino acids. We'll talk about the different structure levels of proteins here momentarily, but the structure is generally weak and they can be broken, and this can result in what's called being denatured or denaturization, and that's essentially when the protein is broken. It's broken apart and it doesn't return to its former structure, and so then that would make the protein unusable. It's essentially broken. And temperature and pH, we'll see pH in a second. Temperature is our first example. And enzyme activity, so enzymes are types of proteins, and enzymes carry out reactions to lower the amount of energy that's required to carry out that reaction. Enzyme activity increases with increased temperature, and as temperature increases, that causes molecules to move around faster. And enzymes and what they react with, their substrates, are essentially just molecules moving around, and as they bounce together and they connect, that reaction is able to take place. If those molecules are moving around faster because of increased temperature, then there's a faster rate at which that reaction will take place, and so it happens faster. This is up to a certain point, and we can see that in the graph here. As the temperature increases, the rate of activity of that enzyme also increases up to this point of where it's denatured because it's too hot and the protein denatures and it stops working. And so essentially the too much heat destroys the bonds in the enzyme, it causes it to denature. The level of temperature of where this takes place is different for every protein. And so not every protein is going to function within the same temperature range. That varies depending on the type of protein, but typically temperature helps to speed up reactions up to a point, which then there's a quick drop off because that protein is being broken apart, denatured. We see a somewhat similar reaction occurrence take place with pH. Depending on the enzyme, each enzyme and each protein has an optimum pH that it's able to function in. For example, enzymes and proteins that are in our stomach to help to digest food, those can withstand very acidic conditions because the stomach acids help to break down the food. And so the optimum pH for those enzymes is going to be very different from other enzymes that are not functioning in acidic environments. And so whether it's acidic or basic, it can cause the enzyme to denature, breaking of the ionic bonds in the R groups if it moves too far away from the optimal pH value. And so we can see that in our graph here. If the optimum pH is 8, as it moves to be more basic or to more acidic, the enzyme activity decreases to a point where it eventually would denature, the protein would denature. And pepsin is a good example of this. That's the enzyme in our stomachs that can withstand really low pH values. The 20 amino acids that make up proteins vary in their chemical nature due to the R groups, what's attached at that middle carbon in the backbone. And the R groups help to determine the chemical characteristics of the amino acids, whether they're hydrophobic or hydrophilic. And it depends on what's attached there. And so we have some that are nonpolar, some that are polar, some that are basic, some that are acidic, and it just depends on what's attached at that carbon. Sometimes amino acids can be modified after the synthesis of the protein. Collagen, for example, contains the amino acid proline. Sometimes some of it can be converted to hydroxyproline, which makes the collagen more stable. And so the amino acids are diverse in the different types because of what's attached to the R group, and this then gives it different chemical characteristics that are important for membrane proteins we'll discuss in a moment. So now we'll focus on the structure of proteins. And the primary structure, the first level, is simply the sequence of those amino acids that are linked together by peptide bonds. And the backbone of the proteins is the sequence of the two carbons and the nitrogen that are linked by covalent bonds. So the carbon-carbon-nitrogen that's connected to another carbon-carbon-nitrogen, that is what makes up the backbone of proteins and this primary structure. The bond angles of this connection of the carbons and the nitrogens are tetrahedral and can rotate, and this allows the folding into pretty much any 3D shape. And so after the protein is synthesized, typically they're folded into a particular shape, most of them are, that helps it to then be able to carry out its function. And this folding into a particular shape is based on the amino acid sequence itself and the R groups and their attractions or hydrophobic, hydrophilic properties. The amino acid sequence, so how we get the sequence of amino acids in the correct order, is determined by the DNA, and that the DNA, the gene specifically, provides instructions for folding to make that specific sequence, again, based off the R groups. This 3D arrangement of the atom, so after it folds into the polypeptide or protein, is its conformation shape. And the conformation helps to determine what the function of the protein is. Our secondary structure level is all based off of folding. And after the formation of these peptide bonds between two different amino acids, the carbon and double bonded oxygen and the nitrogen with its hydrogen attached that remain from the carboxylic and the amino amine groups, the oxygen is slightly negative and the hydrogen of the nitrogen is slightly positive, and so this creates a little bit of polarity. And what this allows is for hydrogen bonds to be able to form between these two different groups. They're generally weak, but a large number of these hydrogen bonds helps to create stability. And so this results in two different shapes. The first is called an alpha helix, and this is kind of like a double helix of DNA, except it's just one strand, so it's one kind of spiral staircase. And it's a helical shape with hydrogen bonds between the adjacent turns of the helix. The second possibility is called a beta pleated sheet, and this is two or more sections of polypeptides that are arranged in parallel with hydrogen bonds between. And the sections run in opposite directions, and it forms kind of a folded piece of paper or folded sheet with little peaks and valleys if you were to fold a piece of paper kind of accordion style. And so you should be able to recognize these. If you're given a 3D image of a protein, you wouldn't be asked to name the different parts, but you should be able to recognize, oh, that's an alpha helice or that's a beta pleated sheet, and both of these are held together by hydrogen bonds. Our tertiary level or third level of structure is all based off of the folding of the protein. And for some proteins, this is the end of the structure line. They stop at tertiary level. And so this is when that sequence of amino acid, that chain of amino acid gets folded into its 3D shape. The structure is stabilized by the interactions between the different R groups. And there's four different types of possibilities, as we see here. The first being ionic bonds between the positive and negative R groups. We can also see hydrogen bonds between the R groups. We can see disulfide bonds between the pairs of cysteines, and this forms covalent bond, and this is the strongest bond of all of them. Or we can also see hydrophobic interactions between any of the non-polar R groups. And so those are the four different ways that interactions between those R groups take place. This structure and this folding is developed as the protein is being synthesized. These bonds and these interactions occur as the structure is being synthesized. And it allows for many different shapes, most of which are globular, and we'll talk about those in a moment. Some are not folded at all, and they remain as elongated chains, and this creates fibrous proteins, and we'll talk about those also in a moment. And so this helps to further create stability within the protein and fold it so that it can carry out its particular function if it does have folding. With tertiary structure, it's important to discuss a little further the polar and nonpolar properties. For nonpolar, as we previously have discussed, that's going to create hydrophobic conditions, whereas polar is hydrophilic conditions. And so many of the proteins are embedded within the membrane of the cell, the plasma membrane, and need to have some specific properties in order to be able to function. And so many globular proteins need to function in an aqueous solution. This would be like outside of the cell, external to the cell, or internal within the cytoplasm. And so they must have hydrophilic amino acids on the surface where there's the contact with the water. However, then they also need to, if they're transmembrane proteins, if they go across the membrane, they also need to have hydrophobic amino acids in the middle. And if it meets these requirements, if it has hydrophobic in the middle and hydrophilic on the outside amino acids, this helps to stabilize the tertiary structure by maximizing those hydrophobic interactions in the center of the membrane and the hydrogen bonding between the hydrophilic and the aqueous solution. So it helps to stabilize it, actually. Non-polar substances, so those that are hydrophobic, require amino acids to also be hydrophobic. Otherwise, they wouldn't be able to function with one another. And so integral proteins are those that are embedded within the membrane, as well as transmembrane proteins in areas of hydrophobic regions. These stabilize the tertiary structure of the protein and correct the positioning to allow it to be able to function. And so depending on where the membrane is found is going to have some then specific requirements of whether or not those amino acids are hydrophobic or hydrophilic. This creates some very beneficial conditions, because then channel proteins that are across the plasma membrane, it's essentially a tube or a channel, it allows hydrophilic solutes or water or substances to diffuse across the hydrophobic core of the membrane. So we've talked about phospholipids. The tails, the fatty acid tails, are hydrophobic. They don't interact with aqueous solutions. And so hydrophilic solutes or water have difficulty being able to move across the membrane. The channel proteins then allow those molecules, those types of molecules, to be able to move across the membrane. And those hydrophilic regions of the protein help to hold it in position and keep it stabilized within the membrane. So we've arrived at our final level of structure, the quaternary structure. And some proteins are a chain of single polypeptides. This goes all the way up to tertiary structure. However, others can be two or more polypeptide chains put together, and also sometimes include non-polypeptide components. And so this quaternary structure is the 3D arrangement of these different subunits, units put together. And there's two different types. Non-conjugated protein would be formed by the linking of polypeptides by the same interactions that we see at the tertiary level. Two examples would be insulin, which has two different polypeptides that are linked by disulfide bonds, so really strong bonds, as we've talked about. Collagen would be another example where there's three polypeptides that are wound together, and these are actually just chains that are wound together, and it creates a really strong structure. The opposite of non-conjugated then is conjugated proteins, and these have one or more non-polypeptide units in addition to the polypeptide. So it's got the polypeptide chains as well as some non-polypeptide, non-protein components. And hemoglobin is a great example of this because it consists of 4 polypeptide chains. We can see this in the image here. There's 4 different components, and it also has non-polypeptide heme groups. And these are chemical structures that's made up of organic compound called porphyrin, as well as an iron atom. And so the oxygen is what binds to the iron atom. The iron atom within this heme group allows the oxygen to bind, and then the hemoglobin that's within the red blood cells can carry that oxygen to the different parts of the body after being oxygenated in the blood. The inclusion of these non-polypeptide components like heme increases the chemical and functional diversity of proteins. So it just opens up more possibilities of how proteins can function. So to finish up, there are 2 main types or structures of proteins that we see. The first is fibrous, and these would be proteins that are elongated polypeptides that lack folding. They don't have that folding structure. They also don't have any secondary structures, the alpha helices and the beta pleated sheets. And so the structure is developed by linking together chains into narrow fibers or filaments through hydrogen bonds. And collagen is a good example of this, where there's 3 polypeptides that are wound together in a triple helix, has really high tensile strength, it's really strong. And the R group of the amino acid faces outwards, and it allows for a variety of variations as well. So we see diversity of variations here as well. And collagen would be a really strong molecule that is obviously very important to humans. Globular proteins would be the other type of protein class, and this has a rounded shape. that's made by the folding. It's stabilized between the interactions between the R groups, so we're seeing tertiary structure here. And the position of each atom is critical to the protein's function, its conformation. Insulin would be an example, is the only molecule that has the correct conformation to bind to its particular insulin receptor site. And so where each of those atoms are positioned within the amino acid, if they're not in the correct places, then it doesn't allow the protein or the enzyme to be able to function. And insulin is an example of that, because the insulin can't bind to its receptor site if the pieces and components are not in the correct sequences, in the correct locations. And so that concludes our discussion and introduction to proteins. We'll talk more about proteins throughout the course, how they function, how they're used. We'll look at enzymes specifically, and we'll see examples as we progress of how proteins can actually carry out all of the amazing, cool reactions that take place within organisms.